Hello guys, welcome to Dr. Science. So today we are going to talk about the metabolism of very low density lipoproteins. So please watch my previous videos regarding the lipid storage and transport in my biochemistry playlist, and this is taken from your Harper's Biochemistry chapter 25. Okay, so please watch my previous videos or else you will not understand a single thing here. Okay, so this is the complex diagram which is taken from your biochemistry textbook, and as you can see here, the metabolic fate of very low density lipoprotein is given here. I will try to make it as simple as possible, but this is a very complex thing to understand. So this is going to be a long video, and please bear with me. Okay, so in order to understand this, we have to draw the structure of very low density lipoprotein. So usually the very low density lipoprotein is going to contain, when I'm talking about very low density lipoprotein, I'm going to talk about the mature form of your very low density lipoprotein. So this is a very low density lipoprotein, and inside the very low density lipoprotein, or the VLDL, the main lipid component is your triacylglycerol. Okay, and not only that, it is going to have apoprotein B100, and this is very important. And aside from that, the mature VLDL is going to also have apoprotein C and apoprotein E. Okay, and these two things are delivered by high density lipoprotein, or the HDL. Don't worry, I will try to explain that one. For a second, you just trust me that the structure of a mature VLDL is going to have apoprotein B100, apoprotein C, and your apoprotein E. Out of this, apoprotein C and E were delivered by high density lipoprotein. Okay, now we are going to see how the very low density lipoprotein is going to get synthesized. I would like to tell that very low density lipoprotein is synthesized in your liver, whereas in the case of chylomicrons, they were synthesized in your intestines. Okay, and very low density lipoprotein will have ApoB100 as their marker, and chylomicrons are going to have B48 as their marker. Okay, these things are very important. Okay, now we are going to see how the synthesis of VLDL will take place in the liver. So this is the liver, and this liver is going to synthesize the VLDL. Now the synthesis of the VLDL which takes place in the liver, usually the liver synthesizes a nascent VLDL. By nascent I mean this VLDL is very immature. Why it is immature? Because it have only one apoprotein present on its top. And what is the apoprotein that is present on it? It is apoprotein B100. Okay, and inside the main lipid component is your triacylglycerol. Okay, so once again, so this is the nascent VLDL, or the immature nascent VLDL, which was synthesized in the liver. The main lipid component is your triacylglycerol, and it was having only one apoprotein, which is your apoprotein B100. and this nascent VLDL was synthesized in your liver, right? Eventually, this nascent VLDL will get two lipoproteins. It will get two lipoproteins, and that is from HDL. So we can say that HDL is a good friend of VLDL, because HDL, usually the structure of HDL, it contains of mainly three lipids, apoproteins. Okay, it consists of three apoproteins, which is your apoprotein A, apoprotein C, and apoprotein E. Remember mnemonic ACE. HDL will have apoproteins A, C, and E. Okay, so out of these three apoproteins, the HDL, since HDL is a very good friend of the VLDL, it will donate two apoproteins, which are your apoprotein C and apoprotein E. So here is your HDL, which lost your apoprotein C and E. So it will have only one apoprotein, which is your apoprotein A. Okay. So eventually, after receiving apoprotein C and E from HDL, there will be formation of your mature very low density lipoprotein. And as I already said you, the main lipid component is your triacylglycerol. And what are the apoproteins present on it? Apoprotein B100, and the two apoproteins which were delivered by HDL were your apoprotein C and apoprotein E. So in my previous videos, I already told you that apoprotein C is required by lipoprotein lipase, and apoprotein is required for destruction in the liver. Don't worry, I will explain this one briefly, because in my previous video I already explained about the receptors apoprotein C and apoprotein E and their importance, and in this video I'll just give a brief introduction about it. Okay, so eventually this mature VLDL, I'm going to draw this entire thing again. So let's copy this one, so it will be easy for us. Okay. Okay, my pen is not working. Let's copy this one here. Copy and paste. Okay, so as you can see here, so this was the mature VLDL. Okay, so upon this mature VLDL, there is an enzyme called as your lipoprotein lipase. Now, what is the function of this lipoprotein lipase? I will tell this one very briefly, so it will be clear for every one of us. So we will draw the structure of mature VLDL, and this mature VLDL, the main component was your triacylglycerol. Okay, this was triacylglycerol. So usually here is your glycerol. What is this? Glycerol. Okay, first I want to draw the structure of lipoprotein lipase. So this is the endothelium. On this endothelium, we have heparin sulfate. What is this pink color thing? This is your heparin sulfate. On this heparin sulfate, there is a enzyme called as your lipoprotein lipase. Now the function of this lipoprotein lipase is to break down the triacylglycerols. So as you can see here, this is the mature VLDL. Okay, eventually the mature VLDL will have apoprotein C. Let's see here, the mature VLDL will have apoprotein C2, especially the type 2 receptor or the type 2 apoprotein. So the apoproteins, I'm going to call it apoprotein C, so it will be easy for us. Okay, so apoprotein C is necessary for the enzyme lipoprotein lipase. If there is no apoprotein C, the lipoprotein lipase cannot extract the triacyl glycerols. Okay, so in simple terms we can say that apoprotein C is required for lipoprotein lipase enzyme to work. In its absence, it cannot metabolize the triacyl glycerols. Okay, so once again, here is your apoprotein C. So once the apoprotein C and lipoprotein lipase interaction takes place, the triacyl glycerol, I am going to draw the structure of triacyl glycerol. This is the glycerol component, and to this glycerol there are 3 fatty acids. There are 3 fatty acids. So this entire compound is called as your triacyl glycerol. So eventually the triacyl glycerol by the enzyme lipoprotein lipase was getting converted into glycerol and fatty acids. Okay, and these fatty acids will eventually get stored in the adipose tissue. Okay, eventually the adipose tissue will store the fatty acids. So that was the function of the lipoprotein lipase enzyme. So lipoprotein lipase enzyme was breaking down the very low density lipoproteins which were having triacyl glycerols present in them into glycerol and fatty acids. The fatty acids were stored in the adipose tissue. I hope you are clear with this. Next we are going to see what is the next step in the metabolism of very low density lipoprotein. So this was the mature form of very low density lipoprotein, and it was having ApoB100. And what are the two apoproteins? Apoprotein C, which was required for the enzyme lipoprotein lipase, and the other apoprotein was your apoprotein E. Okay, so eventually on this mature VLDL, the enzyme lipoprotein lipase will work, and during this step what was happening? Triacyl glycerols were getting extracted from the very low VLDL. Okay, triacyl glycerols which were present in the stomach of the VLDL were getting extracted by the enzyme lipoprotein lipase. And not only that, during this step the apoprotein C will also get extracted by the enzyme lipoprotein lipase. Okay, so the newly formed compound is called as your intermediate density lipoprotein. So VLDL is going to get converted into IDL, very intermediate density lipoprotein. As I already told you, this thing is going to have ApoB100, and not only that, it is going to have apoprotein E. As I already said, the apoprotein C will get ejected by the lipoprotein lipase enzyme, and this apoprotein C will go and stick to the HDL which donated the apoprotein C and E. Okay, but I am not going to mention this one here, so it will be easy for us. Okay, not only that, the intermediate density lipoprotein will have little bits of triacyl glycerols and cholesterol as their lipid content. I hope you are clear with this. And eventually this intermediate density lipoprotein will get converted into a compound called as your low density lipoprotein. And during this step, once again, apoprotein E will get extracted. Okay, so what is the only apoprotein present on the LDL or the low density lipoprotein is your apoprotein B100. Okay, and what are the lipid storage of low density lipoprotein? It will contain only cholesterol. Okay, and this cholesterol is bad for your health. Hence, LDL is considered as bad cholesterol. Okay. Now, the metabolism of low density lipoprotein will take place in the liver. Now we are going to see how the low density lipoprotein will get metabolized in the liver. So this is the liver. Usually the liver have a receptor called as your low density lipoprotein receptor. Now this receptor can recognize two types of apoproteins. One is your apoprotein B100, which is present on your LDL, low density lipoprotein. The other one is your apoprotein E, which is present on kilomicrons. Okay, so low density lipoprotein can only recognize these two types of apoproteins. Once the recognition will take place, that protein will go inside and the metabolism will take place. Okay, don't worry, I will just give you a small example here. So this was the liver, and this liver is going to have LDL receptor, and it can recognize only two types of apoproteins, which is your apoprotein B100 and the other one was your apoprotein E. As I already told you, low density lipoprotein, which is rich in cholesterol, so this is the cholesterol present in it, will have what kind of apoprotein? Let's draw this with a different color. It will have apoprotein B100. So in order for the low density lipoprotein to get metabolized, since low density lipoprotein is going to contain a lot of bad cholesterol, right? So this cholesterol should get metabolized in the liver, or else the cholesterol will get increased in our body and there will be atheroscleromatous plaques in our body. In order to prevent this, the metabolization of the LDL, low density lipoprotein, should take place. Now how this is going to take place? As I already told you, low density lipoprotein will have ApoB100, which is ApoB100 protein present on it. So eventually this low density lipoprotein will go and interact with the LDL receptor. Okay, as I already told you, LDL receptor can recognize ApoB100, and what is the other apoprotein it can recognize? It is your apoprotein E. So low density lipoprotein already have apoprotein B100, so it will go and interact with the LDL receptor, and in the liver it will get destroyed. The low density lipoprotein will get destroyed. Okay, so once again, very low density lipoprotein have two apoproteins, which is your apoprotein B100, apoprotein C, and apoprotein E. Eventually, by the enzyme lipoprotein lipase, the lipoprotein lipase will extract the triacylglycerols, and not only that, it will extract your apoprotein C. The newly formed compound is called as your intermediate density lipoprotein, which was having B100 and apoprotein E. So the liver can recognize these two apoproteins. Okay. And finally, intermediate density lipoprotein will convert into low density lipoprotein. Low density lipoprotein. And during this step, the apoprotein E was getting extracted. So what we are left with is your apoprotein B100. Okay. So please take a notes of this one. This is very, very important. Don't worry, I will draw the entire diagram once again so it will be clear for us. So this was the liver, and liver was synthesizing nascent or the immature VLDL, which was having triacylglycerol as the main component. On the top of it, it was having ApoB100 protein. So this is your nascent VLDL. Eventually, it was getting converted into mature VLDL. What is this? This is your mature VLDL. What is the main lipid component? Obviously, it is triacylglycerol. Okay, and HDL, which was having three apoproteins, apoprotein A, apoprotein E, and apoprotein C and apoprotein E, was donating two apoproteins, which are your apoprotein C and apoprotein E, to the mature to the nascent VLDL. Okay, so once again, the mature VLDL will have B100, ApoB100, apoprotein C, and apoprotein E. Okay, so here is your HDL, the remnant HDL, which will have only apoprotein A, since the C and E were donated to the nascent VLDL. Okay, to this mature VLDL, there is an enzyme called as your lipoprotein lipase, and this lipoprotein lipase will recognize the apoprotein C2, or we can simply call as your apoprotein C. And once it recognizes the apoprotein C present on the mature VLDL, it will extract the triacylglycerols, and not only that, it will extract the apoprotein C, and this apoprotein C will travel and get deposited on the HDL. Okay, now the newly formed compound is your intermediate density lipoprotein, which will have little bits of triacylglycerols and a little bits of cholesterol present in it. Okay, and on the top of that, it has ApoB100 present on it, together with apoprotein E. Since apoprotein C will get, as you can see here, apoprotein C will get extracted by the lipoprotein lipase. So only apoprotein E will be present in the intermediate density lipoprotein. Okay, and finally, intermediate density lipoprotein will get converted into low density lipoprotein, which have only cholesterol as their lipid component. And whenever IDL is getting converted into LDL, during this step, apoprotein E is also extracted, and this apoprotein E will go and get deposited on the HDL. Okay, so the LDL will have only one apoprotein, which is your apoprotein B100. Now eventually the metabolization of the LDL will take place in the liver, and this liver will have a receptor called as your low density lipoprotein receptor. and this receptor will recognize the B100 apoprotein, which is present on the low density lipoprotein, and once it is recognized, LDL will get metabolized or destroyed in the liver. Okay, so that was about it, and don't worry, I will just explain the pathway here. So please take a look at here. So this is the nascent VLDL. This is the nascent VLDL. This is the nascent VLDL, and nascent VLDL will have ApoB100 present on it. Okay. So from the HDL, as you can see, this is the HDL, which contains apoprotein A, apoprotein C, and apoprotein E. The HDL will donate apoprotein C and apoprotein E to the nascent VLDL, and it will get converted into mature VLDL, which will consist of ApoB100, apoprotein E, and apoprotein C. Okay. The enzyme lipoprotein lipase will extract the triglycerols from the mature VLDL, and it will get converted into intermediate density lipoprotein. And during this step, apoprotein C is extracted and get deposited on the HDL. Now the intermediate density lipoprotein will have only ApoB100 and ApoE present on it. Once that step is done, the IDL will get converted into LDL, and during this step, the apoprotein E will go and get deposited to the HDL. Now the low density lipoprotein will only have ApoB100 present on it, and as you can see here, the LDL receptor will only recognize ApoB100 and ApoE apoproteins. Okay, and once it recognizes the B100 present on the LDL, it will get metabolized in the liver, where the final destruction of the LDL will take place. Okay, so that was about the metabolism of low density lipoprotein. I know you are a little bit confused because this is a very tough topic to understand, and not everyone will understand in a single go. So please watch it again and again, 2-3 times if you want. And once it is done, please refer to your textbooks, then you can understand it properly. Thank you guys.